Bacteria are commonly found in the marine environment. Most bacteria are not harmful, however, some are known to cause diseases. These bacteria are usually present in small numbers, but may proliferate in hatcheries where aquatic animals are maintained at high densities. These bacteria are referred to as opportunistic. The most commonly occurring bacterial disease in marine bivalve hatcheries is vibriosis. Routine monitoring for Vibrio is important in bivalve hatcheries, especially during spring and summer when temperatures rise, causing conditions favorable to bacterial growth. In this video, we share techniques used for on-site hatchery monitoring of Vibrio, including media preparation, sample collecting, sample plating, and interpreting results. The media of choice for isolating Vibrio is thiosulfate citrate biosalt sucrose agar, or TCBS. Ready-made plates can be purchased from scientific supply companies such as Fisher Scientific, or powdered media can be purchased and plates made on site as needed. Plates can be refrigerated up to a month. Just remember to bring to room temperature before using them. In addition to the TCBS media, you will need a sterile pipette that dispenses 1/10 of a milliliter, sterile spreaders, parafilm, and a Sharpie for labeling plates. These supplies can be purchased from online companies such as Amazon. If you need to make dilutions, you will need additional supplies, such as sterile 1.5 milliliter centrifuge tubes and sterile seawater. If making your own media, you will need a balance to weigh the media, a hot plate with a stirrer, a stir bar, a glass beaker or flask, a graduated cylinder, petri dishes, and heat-resistant gloves. To prepare the TCBS agar, follow the label directions. The number of agar plates needed will determine the amount of media to prepare. Approximately 15 milliliters of liquid media is needed for each plate. Measure the amount of distilled water needed using a graduated cylinder. Weigh out the appropriate grams of powdered media. Pour the water into the flask. Add the powdered media in a stir bar. Use a flask that holds twice the amount of media needed. This is important as the media will be brought to a boil after mixing. Turn on the stir plate to mix the media first, then add heat. Allow the mixture to come to a full boil. Remove the flask from the hot plate using heat-resistant gloves immediately after the mixture comes to a full boil to prevent the mixture from flowing out of the flask and onto the hot plate. To prevent bacterial contamination, wipe down the work area where the plates will be prepared with a 5% bleach solution and disinfect hands or wear disposable gloves. Allow the flask to cool before pouring the agar. The flask needs to be cool enough to hold while pouring, but warm enough to prevent the agar from solidifying. Remove the number of petri dishes needed from the sleeve and lay them out on a table or bench. Carefully pour enough liquid into each dish to cover the bottom. Replace the lids. Let the plates sit overnight at room temperature to allow the agar to solidify. If not using the plates immediately, place solidified plates in sealed plastic bags and refrigerate them upside down. This will prevent condensation from occurring. There are many locations in a hatchery that should be routinely monitored for Vibrio, including incoming source water, treated water, larval and broodstock tank water, and algae cultures. Algae cultures should be sampled at various points, including test tube inoculums, flasks, carboys, and bags or sun tubes used to feed the clams. Disinfect your hands with alcohol prior to collecting samples, or wear gloves to prevent contamination of collected samples. Use a clean, sterile container to collect the samples. Label the sample collection container with a Sharpie so that samples added to TCBS plates can be easily identified with respect to collection time and location. Clean the work area with a 5% bleach solution prior to adding the sample to the plate, and disinfect hands with alcohol or wear gloves. Gather needed supplies such as pipettes, spreaders, collected samples, and prepared TCBS plates. Use a Sharpie to label the bottom or agar side of the plate with a sample description and the date. It is important to label the plate along the outer rim so that any bacterial colonies that grow can be easily seen and counted. Remove the lid of the Petri dish. Add 100 microliters or 1/10 of a milliliter of the collected sample directly onto the plate using a sterile pipette. Note that this is an automatic 1:10 dilution. Use a spreader to distribute the sample evenly. If it is necessary to further dilute the collected sample, perform a 1:10 dilution by adding 100 microliters of the sample to 900 microliters of sterile salt water. Mix the sample well before adding the sample to the plate and spreading. Agar has a consistency of hard gelatin. Do not press down too hard or you will break the agar. Replace the lid, seal the plate with parafilm to keep it from drying out. Incubate the plate upside down at room temperature. Bacterial colonies that grow on TCBS agar will be either blue-green or yellow. Blue-green colonies represent pathogenic Vibrio strains, whereas yellow colonies represent non-pathogenic strains. Examine the plates at 24 and 48 hours and note which plates have colonies. It is common to find Vibrio in some areas of the hatchery. Disinfected water should be Vibrio-free. Algae test tube cultures and algae flask should be Vibrio-free. Note that the algae collected from the sun tube, used to feed the broodstock tank in this video, has yellow Vibrio colonies. To identify the source of this contamination, the next step would be to sample other sun tubes as well as algae carboys, flask, and test tube cultures. Expect to find some Vibrio in larval and broodstock tanks. Ideally, the number of yellow colonies should be less than 1,000 colony-forming units per milliliter, and the number of blue-green colonies should be close to 0. Count the total number of colonies as well as the number of yellow and blue-green colonies. Note that over time, yellow colonies will turn green. This will also occur if plates are refrigerated. It is therefore important to note colony color when colonies first appear. This can be done by circling or labeling green colonies so that they can be differentiated from yellow colonies later. Multiply the counts by the dilution factor. For example, if there are 20 total colonies and 100 microliters or 1/10 of a milliliter of water was placed on the plate, the total number is 200 colony forming units per milliliter of water. If there are too many colonies to count, collect another sample, replate, and reincubate a diluted sample. Monitoring the water used in hatchery at specific points will alert you to the need to clean reservoirs, hoses, and replace or clean filters and UV bulbs. Contaminated algae fed to larval tanks can affect larval production. Periodic sampling for Vibrio in bivalve hatcheries is a valuable tool that allows managers to identify sources of contamination and take the necessary steps to control or eliminate it, thereby improving clam seed health. This research was supported by the Florida Sea Grant College program and was a collaboration between University of Florida and Florida Atlantic University.